but I'll take this time and introduce Professor Juha Silvanto. So we're delighted to have Professor Juha Silvanto with us today in our Bar-Ilan University Vision Science Seminar. Professor Silvanto did his BSc in psychology at Cardiff University and then continued to do a PhD in psychology at UCL's Department of Psychology and Institute of Cognitive Neuroscience under the supervision of Vincent Walsh and Eli Levy. He then did a postdoc at Harvard in Professor Alvaro Pasquale Leone's lab, and then another postdoc at UCL's Institute of Cognitive Neuroscience. He then got a professor of psychology position at Helsinki University, where he was also a co-director of the Cognitive Brain Research Unit and the Institute of Behavioral Sciences. He led research groups in Aalto University, Finland, and then as a professor in cognitive sciences at the Department of Psychology at University of Westminster in London. For the last couple of years, his lab is located at the School of Psychology at the University of Surrey. Juha Silvanto is an editorial board member of Neuroimage. He published almost 100 publications in peer-reviewed journals, including top leading journals as Nature Neuroscience, Current Biology, Trends in Cognitive Sciences, and Trends in Neuroscience, and others. He has an H index of 41 with thousands of citations. His work is continuously funded by prestigious funding bodies, including the ERC, ESRC, and others. On top of his university teaching and administrative roles, grant reviewer on many prestigious grant panels, being a chief editor at Frontiers of Consciousness Research, he received acknowledgements and a certificate of peer reviewing excellence from multiple journals. His research focuses on understanding the neural basis of visual processes, including motion perception, symmetry, consciousness, working memory, and imagery, and also the methodology of brain stimulation, developing new and effective protocols as state-dependent brain stimulation and others. In his research, he uses TMS and TDCS for brain stimulation and neuroimaging techniques as fMRI and EEG. So without further ado, I'm delighted to invite Juha to tell us about your research. Thanks for joining us. And now I hope that, yes, you're here. Okay, so wait. So I made you a co-host and I've already introduced you. So I hope
we can't hear him.
Yes, we can hear you.
No, I'm not sure.
No.
No.
Sorry, Vadim, it's you. It's not Juha. Wait, I'll pause this.
To share my screen.
We don't see that yet, but you can.
But I can't. I don't think I can't. I can't share it. I don't have the option.
You can. Wonderful.
Okay. Sorry about all this. Okay, I'm going to start. Okay. Well, thanks, Sharon, for the invitation. Obviously, it would be nice to be there in person, but maybe another time. Right. So I'm going to talk to you about one of the sort of topics in neuroscience which has been very controversial over the years, namely the role of the primary visual cortex in generating conscious experiences. And in particular, I'm going to focus on blindsight, which as a phenomenon has also been very controversial, but very important in guiding the way we think about the neural basis of conscious experience. So when we think about awareness, so one model which has been very, one sort of principle of brain organization, which has been very influential in guiding the way we think about the brain and think about awareness, is this model of hierarchical organization of the visual system. So this is the famous model by Van Essen and Monsell, which basically arranges or organizes different brain areas in terms of the feed-forward connections and feedback connections. We basically originate and terminate in different cortical layers, and then depending on whether an area sends feed forward or feedback connections to other areas, we can have this organization of the visual system. Now, this is kind of like an anatomical way to organize it, but it has been very influential in the way we think about conscious experience as well. Can't hear me very well? Okay. And coupled with the anatomical hierarchy, the other feature which has been very important is the functional specialization. So the idea that when we move higher up in the visual system, brain areas become much more selective in terms of the type of stimuli they process. So in the lower levels, V1, we have selectivity for edges and lines, the sort of things. When we move up higher, we have areas like MT and V4, which are selective for motion and color. And then we go even higher in the hierarchy, and we get selectivity for objects and faces. And this increased specialization as we move up to the hierarchy kind of gave rise to the view that conscious experience also has this hierarchy, and that conscious experience reflects the activity of these sort of higher level neurons. So areas higher up in the system are the areas which give rise to conscious experience. So what happens if we lesion different areas of this network? Well, if we lesion the area at the bottom of the network, the primary visual cortex, we basically abolish all vision in the corresponding part of the visual field. So we call cortical blindness, and that's perhaps expected. If we basically cut out all the input from higher level areas, which V1 provides, then it's perhaps not surprising that awareness is lost. But what's interesting about these patients is that even though they have no phenomenal awareness, they are nevertheless able to detect stimuli in their blind field using force joy measures. And there's really a large literature on these from people like Weiss, Grantinel, and Cowie, who have looked at all the different features that can be encoded in the absence of primary visual cortex and in the absence of awareness. And the reason why blindsight is really important for us is that it basically shows the dissociation between visual processing and visual detection and visual awareness. So in the absence of V1, we can still be aware, so we can still detect visual information, even though we are not aware of that information. And this phenomenon kind of changed the view how we think about primary visual cortex as being a region which not only processes information and feeds information forward to higher level areas, but it's also important for more sort of higher level sort of processes related to conscious experience. In practice, how blindsight is reflected is using signal detection measures. So if you have a force choice task, it's also normal in control subjects. So the performance in force choice task goes very closely together with performance in a yes/no task. But in blindsight, so this is the famous blindsight patient GY, you have a dissociation where the sensitivity in a yes/no task is much lower than in a force choice task. Now, one sort of controversy in the field has been the nature of conscious experience in blindsight patients. So there are some people who have argued that actually, even though that these individuals do have a limited amount of awareness, so people like Barber and Semyaseki have argued that they kind of have awareness of a moving stimulus. Whereas there's another sort of other camp in the field who argues that there's no awareness at all. And that's of course very important, because if we want to make conclusions about the role of primary visual cortex in awareness, then the nature of awareness when this area's lesion becomes very important. When you actually ask the patients themselves, this is what they say. So they say things like, you don't actually ever sense anything or see anything. It's more of an awareness, but you don't see it. It's a sense that I haven't got. So I think from this description, it's probably quite clear that the awareness that blindsight individuals have is not really visual, it's not really phenomenal. It's an awareness of something happening, some experience of something happening, which is probably not surprising given that the stimulus does have a physiological consequence in the brain, and somehow probably the patient has learned to kind of associate that physiological consequence with the stimulus. But the main point here is that the awareness is not really phenomenal per se. It's not visual in the sense that it is for us. We don't have these lesions. But there's also been lots of imaging work looking at which brain areas are involved in blindsight. So when you present a motion stimulus to blindsight patients, what you see is activation in motion areas of the cortex. So obviously, the information can reach these areas in the absence of V1. And there's lots of ways which they can reach this. So there's one pathway which bypasses V1 and projects to the extrahead cortex. So clearly, physiologically, it is possible for information to reach these high-level areas. And the point is that even though information can reach extrahead cortex, and even though the patient is able to detect that information unconsciously, this information does not reach awareness. So this would suggest that there's something more to the role of primary visual cortex than merely feeding information forward to higher level areas. And one sort of model which became very influential as a result of blindsight is the reverse hierarchy model. So one famous model was put forward by Victor Lame, who argues that for visual awareness to arise, we need feedback activity from the extrahead cortex back to the primary visual cortex. So in the absence of this feedback activity, information, so with feed-forward activity, information can, visual stimuli can be detected, but we can't become aware of them. So this was the sort of model that resulted from blindsight, and subsequently there were also lots of studies using TMS which demonstrated or supported the view that you need these feedback connections for conscious experience to arise. And one field, one approach which has been quite sort of useful in this research has been the induction of phosphines by the use of transcranial magnetic stimulation. So basically, when you apply TMS over the visual cortex, you induce these flashes of light, which basically result from the direct stimulation of the neurons, which are encoding visual stimuli. And the key point about these phosphines is that they reflect the underlying properties of the stimulated area. So if you stimulate the motion area MTV5, these phosphines contain motion information, and they're also quite large, reflecting the large receptive fields in MTV5. If you induce phosphines from V1, the phosphines are much smaller, they don't contain motion. So basically the phosphine appearance tells you about which area you're stimulating. Now, if it's the case that you need these feedback connections from extrahead cortex to V1 for awareness to arise, then the prediction that arises is that if you stimulate the intact extrastriate cortex of a blindsight patient, you shouldn't be able to induce a phosphine because in the absence of V1, these feedback connections cannot arise. And that's exactly what has been found. So this is what Covey and Walsh found. If you stimulate the intact hemisphere of the blindsight subject, you induce phosphines as you would in a healthy individual. So you induce a phosphine in the contralateral visual field. But if you apply TMS over the intact extrastriate cortex of the damaged hemisphere, so the red area here shows the lesion, you don't induce phosphorylates. So even though this area is able to process information, and this area, the activity in this area underlies blindsight performance, this activity is unable to reach awareness in the absence of primary visual cortex. So this was further strong evidence for the view that extrastriate activity does need these feedback connections to reach conscious experience. There's also studies then with healthy individuals and using visual stimuli, which kind of show the same principle, that basically you have these information. So for us to become aware of motion information, we need these feedback connections from V5MT back to V1. So with TMS, what you can do is you can trace the information flow through the visual cortex by applying stimulation at different time windows relative to the target onset. And what you find is that when you apply TMS over the primary visual cortex, you have a late period, you have an early period activity at which you can disrupt performance, presumably reflecting the initial feed-forward flow of information. But you also have a late period of activity in which TMS impairs performance, and that presumably reflects the feedback activity. So basically, at this point, it looks like really the primary visual cortex is very important for awareness. If it's lesioned, you lose conscious experience, and even if you directly stimulate extrastriate areas in these patients, you don't induce phosphins. In normal participants, the timing of effects induced by TMS also indicate that this feedback activity from extract areas to V1 is necessary for awareness to arise. But actually, I mean, the situation is slightly more complicated than that. And I'm going to talk about some studies now where we use brain stimulation to trying to induce conscious experiences in patients without V1. But before doing so, what I'd like to mention is that there's actually quite a long history of visual experiences in the blind field of blindsight individuals, which haven't really been, I guess, really taken into account in the sort of theoretical thinking about consciousness. So there's this phenomenon of hemianopic completion, which has been known for a long time, since the time of Popper-Reuter, where basically, if you present a stimulus, which includes both the intact visual field and the damaged visual field, the patient can actually perceive the whole stimulus. So if you present the stimulus only within the hemianopic field, the patient cannot consciously experience it. But if you present it in a manner that some of it falls into the normal field, then the patient can actually perceive the whole stimulus. So what this indicates that there's probably more to our awareness than simply these feedback connections, because obviously in these individuals, the part of V1 which encodes the hemianopic field is missing, yet they are able to experience visual percepts. So that's sort of inconsistent with the idea that you always need the primary visual cortex for awareness to arise. So what we try to do is to use sort of a similar approach, in the sense that we induced activity in both hemispheres at the same time by applying TMS simultaneously over the left and the right side of the extrastriate cortex. So in these individuals, this is the patient GY. He has a lesion in his left visual cortex. So obviously, if you stimulate the left side, the information cannot reach awareness via the left V1, which is missing. But what we try to do is that if you stimulate both left and right V5, so you stimulate bilaterally, perhaps the activation in the damaged hemisphere can reach awareness as part of a percept originating from in the intact hemisphere. So what we did was we applied TMS over the left and right hemisphere over the motion area V5MT in very close temporal proximity. And what we manipulated was basically the time delay between the stimulation between the two pulses. So when we apply TMS over the left and right side at exactly the same time, so there's no stimulation asynchrony at all, he only perceives a phosphene in his good visual field. So these are his drawings. So what happens in this experiment is that you apply the stimulation, then you ask the person to draw what they perceived. However, if you introduce a stimulation onset asynchrony of sort of 20-60 milliseconds, what happens now is that he can actually perceive a bilateral phosphene. So he can actually experience visual qualia in his blind field, even though he doesn't have a primary visual cortex. So what this can demonstrate is that, in fact, the activation in the damaged hemisphere can reach awareness. We don't exactly know how that happens, but obviously the theoretical implication is that, obviously it cannot be the feedback to the ipsilateral primary visual cortex that gives rise to awareness, because that area is lesioned. So obviously there has to be other ways by which visual information reaches awareness. We also did a further study where we tried to see whether this person can experience colored percept as well in his blind field. So this is based on an adaptation paradigm in which, if you adapt a person to a color prior to application of TMS, the adaptation basically modulates the qualia of the induced phosphene. So if you adapt to a uniform red color, you will perceive an after image in the complementary color, so you perceive a green after image. If you then apply a TMS pulse, you induce a red phosphene. So basically what TMS is doing is it's sort of preferentially activating the adaptive neural populations. And what we wanted to do is to use this approach as a tool to probe further how the conscious experiences arise in GY. So what we did was we adapted GY to different types of stimuli. So we could either adapt him to stimulus that that intrudes both the blind field and good field, or we could adapt only the good field or only the blind field, or we could present different colors to the two hand field. Obviously, the question is, does the adaptation of the blind field affect the qualia of the visual stimuli, of the induced phosphene? So when we present a bilaterally red adapting stimulus and we induce a phosphene, GY now perceives a bilateral phosphene. So the phosphene component in both of his hemispheres is red. Now, when we adapt only the blind field, I will present basically this black stimulus to the good field, the phosphene appears without color. So what this shows is that adapting merely the blind field has no effect on the induced conscious percept. And in the final condition, so if you adapt the good field, so here we adapt the good field to green and the blind field to red stimulus, you now perceive a green phosphene. So what this basically means is that it's the adaptation of the good field that determines the phosphene color. So this kind of suggests that it is the activation in his normal hemisphere which underlies the qualities that he can perceive. So, I mean, we don't exactly know how these percepts arise. I mean, we don't know the exact neural mechanisms, but if you think about the theoretical debate, particularly in terms of feedback connections and the role of this sort of recurrent feedback activity between extract cortex and primary visual cortex, well, these results would suggest that there has to be more to the story, because obviously even an individual, the activity in the damaged hemisphere can reach awareness. So this means that there has to be other ways for awareness to arise. Sorry. Can you still hear me?
Yes, we can hear you.
So the question that arises then is, well, what is really the importance of this feedback activity? So in these models of Lame, he's argued that it's really about visual awareness. But there are other models which were sort of developed at the same time, which maybe didn't get as much attention, where feedback was considered to be important for visual processing more generally. So for example, Boulier had this model in which basically feedback connections is important. It basically allows magnocellular activity to guide processing of parvocellular input. So the idea there was that motion information is processed faster. So by the time information regarding things like color is processed in primary visual cortex, you already have feedback from V5MT coming back to V1, which can kind of guide the processing. Also, the other aspect of this model was the fact that with feedback activity, you can have kind of the large receptive fields of exercised neurons. They can guide the processing of neurons in V1, which have smaller receptive fields. So that way, visual processing already in V1 can take into account the context in which a stimulus is presented. So what we tested here was specifically whether this late period of V1 activity in response to feedback is also applies to non-conscious processing. And the way we did this was have the participants to give two responses. First of all, so they were presented with a motion stimulus. It's a left-to-right judgment. So they need to indicate the direction of the stimulus. And the second response is a subjective ratings, how aware they were of the motion stimulus. This is on the scale of 1 to 4. They say 1, it means I did not perceive any motion at all. Response 4 would be I saw the direction of the motion. And then what we looked at was stimuli, the perception of, the performance with stimuli that the participant said that they didn't detect. So perception, so the perception without awareness and perception with awareness. And basically what we find is that in both cases, so regardless of whether the participant says they saw the stimulus or they didn't see the stimulus, we find that this late period of V1 activity is important. So when you apply TMS at these late time windows, you impair performance, regardless of whether they respond that they were aware of the stimulus or they were not aware of the stimulus. So what this indicates is that this late period of V1 activity seems to be important for all visual processing, not only for conscious experience. So basically, I guess the implication there is that probably the view that feedback is particularly important for conscious processing is probably not accurate, but rather it's more likely that we need feedback processing for all visual functions, whether they're conscious or not. And I guess this would take it more to the sort of line of Boulier, Hochstein, who have talked about these reverse hierarchies and how this sort of feedback from high-level areas is important in guiding the visual processing of V1 neurons, which basically, because of the small receptive field size, can't see the context of visual stimuli. That's why the feedback is important. But then, if that's the case, it's still a bit mysterious about why awareness is lost when V1 is damaged. So if V1 simply plays, if these feedback connections are not really important for awareness, but just they enhance visual processing, why is awareness then completely lost after V1 lesion? And why is V1 the only region in the brain which completely abolishes the visual awareness? And I guess one sort of clue comes from the fact that if you look at blindsight performance, how good it is, what kind of stimuli can the patients detect? Actually, the performance is quite poor. So if you look at things like contrast sensitivity, you actually you need to have a very salient stimuli for the patient, for the blindsight patient, actually to detect it. And lots of features which people talk about, things like object discrimination. You know, if you do the studies in a way that you control for low-level factors, you know, these kind of discrimination performance is absent. So the point is that blindsight is actually extremely poor. I mean, the sensitivity, contrast sensitivity, motion sensitivity, all of these are very, very poor. So it's not correct to argue that blindsight is really a dissociation between awareness and performance, because performance is very poor as well. And also, if you look at the neural activity, so if you look at motion responses in the damaged hemisphere, so yes, the motion area MTV5 can be activated, and this activity, you know, does underlie blindsight performance. But if you look at the responses, they're actually very weak. So it's clear that even though there are pathways which bypass the primary visual cortex, the level of activity in MTV5 is really, really poor compared to sort of healthy hemisphere. So I think probably the kind of confusing aspect in the field has been the fact that blindsight has been kind of conceptualized as a pure dissociation between awareness and visual processing or visual detection. But in fact, even though some visual processes remain, they are they're very impoverished. Same with the neural activity, it's very impoverished. The neural responses are very poor. So if you think about theoretically using blindsight as a model of you know, dissociation, you know, that area needs to be reconsidered. So perhaps a sort of more sort of simple explanation is that the reason why blindsight is blind is that V1 might be simply important for enabling normal neural responsiveness in the whole visual cortex. So V1 is the main source of extra input. When this input is lost, the rest of the visual cortex cannot function normally. The neural responses are impoverished. They're very sort of, you know, firing rates are much lower. The selectivity, neural selectivity is lost. And consequently, it could be the case that this explains why awareness cannot arise. So it's not about feedback activity as such. Why V1 is important is simply because V1 lesions has such a detrimental effect throughout the visual cortex. And I think, yeah, and the reason why that perhaps blindsight is not as important for the debate on consciousness is the fact that the dissociation that is often talked about is not as pure as is often described as. So it's not there's no clean dissociation between consciousness and visual processing. So just to summarize, so why then is V1 important? Well, it's a major hub in the visual cortex. It provides most of the input to high-level areas. When this input is lost, the neural responses in the high-level areas are very impoverished. And basically, a simple sort of explanation is that the remaining neural responsiveness may be sufficient for very low-level detection, but it's not sufficient for high-level processes such as shape perception, you know, face perception, visual awareness. So that's a sort of alternative explanation of how why V1 appears to be so important. Okay, so this was my slide. So again, apologies for the trouble in the beginning. I hope you could hear me reasonably. Let me just stop sharing.
So I want to invite everybody to unmute yourselves and give a big applause to Juha, which overcame not only quite amazing obstacles, and yet it was a great, fascinating talk. And I would like to invite the audience, if it's okay with you, Juha, to ask questions.
Yeah, sure, yeah.
Okay, so the stage is open. Rafi, I can see your way. Yeah, Rafi.
Hi, great talk, very insightful. I really liked it. I have one comment, one question. It seemed to me that you suggest that the feedback to V1 is there to enhance resolution or some kind of the thing. I mean, this reverse hierarchy concept. If this is the case, how do you explain the fact that in your phosphine completion experiment, it didn't seem that the phosphine appearance in the hemisphere that was damaged were more vague or more diffuse or more blurry? Wouldn't you expect that because of the lack of proper feedback to early visual cortex? In the drawing, it appeared exactly symmetric.
Yeah, I mean, that's a good question. I mean, the real problem in the problem in the phosphine work is that we don't really know how they rise. So it's really difficult to answer that question. Partly because also the phosphine slightly differs in appearance between participants. But I mean, what
but the same holds also for the completion experiment.
Yeah.
To describe the classic completion experiment.
Yeah, that's a good question. Yeah, I don't know exactly what I would answer.
Ellie Pelly, yeah.
Thank you. You know, I've been wondering for many years why we see blindsight experiment almost exclusively with hemianopia rather than people that had double-sided strokes, which there are plenty of them that are there. You know, so one of the explanations for that has been artifacts in the eye, a visual artifact that goes into the seeing side. But your presentation suggests that maybe it's the existence of the functional V1 that is only enabling such a thing. So is it true that having the V1 on the intact side may, based on your analysis and your interpretation of the results, that this is what is making it possible to have blindsight, that it's necessary for it to have at least one V1 out of the two to provide the conscious, or I don't know if we call it conscious, it's sufficient to response. I mean, even for these subjects, it's not really conscious.
Yeah, I mean, I guess if you had a bilateral damage to V1, then you would have, I mean, the extra. Sorry, I'm just getting a lot of feedback. It's very annoying. I mean, I guess because when you have a unilateral lesion, then you have a one V5 that has normal, I mean, neural connectivity. You have normal neural responses. I suppose if you had a bilateral lesion to V1, then both extrastriate cortices would be, you know, they would have a reduced level of activity, reduced excitability. So I suppose, I mean, I guess the point is that the V1 is maintaining the normal responsiveness of the contralesional hemisphere, which allows the phosphine to be induced. But in fact, I'm not aware of studies where people have tried to induce phosphines in a case of bilateral V1 lesion. I mean, it's actually something that, you know, we wanted to do at the time, but we didn't have the patience to do that, because I think that would be a key test in this literature. Not sure I answered your question.
So you're basically agreeing with my stipulation that if you did it, it's possibly that you will not get the response.
Yes, that would be my guess, probably.
And did you really use GY as well? I mean, is all of that based on GY?
Yes, yeah.
Yeah, it's becoming a problem to have this one person.
Yeah, and I think
providing so much of the data
yeah.
in this area, and this patient is, you know, with all due respect, this is his professional livelihood now. In addition to, you know, they're becoming an expert in variety of things.
Yes, and there's also, I mean, with GY also, I think his experience has changed. So he's developed something what they call this type 2 blind sight, where you're really aware of. He's aware of the stimulus that are presented. He can say yes, it was there, but it wasn't visual. So it's kind of evolved as well.
Yeah.
Obviously, the nice thing about him is that he can do brain imaging. There's the other patient that was studied by Weisskranz quite a lot, who who had some metal in his brain, so he could never be scanned. But yeah, I take your point. Yeah.
Thank you.
Crystal, but before you ask, I just want to say, sorry to buzz in, to Ellie, that a lot of neuroscience is based on single cases such as DF for the 2 pathway and LM for motion perception. So yeah, that's in it. We don't, yeah, but okay, sorry, Crystal.
Still is a problem.
Yeah, I would agree with that, with those statements. I think, I have 2 questions, if I may. But first, I want to thank Juha for an excellent talk and some interesting insights. Gave me some food for thought here. My first question has to do with GY, actually, because with your phosphine completion across into his blind field, is that a unique phenomenon to GY? Given the fact, and I would almost think that it might be, but you can correct me if I'm wrong, because he had his lesion when he was a kid and has unusually strong connectivity with the contralateral intact hemisphere. So it doesn't entirely surprise me that he would have this very ready completion of the phosphines if you stimulate the intact hemisphere. So that is my first question. So maybe I'll let you answer that.
Yeah, so I think there's a group in Italy who have done work with other patients. So I think it was Silvia Savazzi. And what's the name of this Italian guy? But I mean, there are papers out there where they found percepts in other individuals as well.
Marco Tomietto, is it?
Pardon?
Not Marco Tomietto?
It's the name escapes me now. It's a very just on the tip of my tongue. He's a very famous Italian neuropsychologist. Done a lot of work with the blind.
Marzi?
Pardon?
Marzi?
Yeah, that's the one. Yeah.
Okay.
So his group has done work also looking at the EEG induced responses when you induce phosphines with TMS in this. So there are a couple of other examples. But I do take your point, the fact that he was 8 when he had this car accident. And obviously lots of plasticity has happened. So I think one sort of valid criticism that's often made is that, is his brain really normal? The kind of conclusions that we draw from him, do they really apply to sort of normal unleashed brain?
So, okay. My second question had to do with the fact that I think it was Arash Sarai who showed that people who have not only a lesion of V1, but also of the LGN do not exhibit blind sight. And so in your circuit diagram, just a thought perhaps, and I would love to hear your thoughts about the potential role of this center. Is it all really cortical? Is everything that we think of as important for awareness rooted in cortex? And could the subcortical centers play an important role in this? And should we really be thinking about them?
Do you mean in terms of the work showing that when you lesion LGN, you
don't get blind sight?
There's all studies from the 90s which show that if you remove superior colliculus and so on, you will abolish blind sight.
Right. I mean, and these subcortical centers also get feedback from extrastriate
Yes.
cortex, right?
Yeah.
So including the LGN.
Yeah. No, I agree. I mean, I think it's I mean, that's a possibility. I mean, I think my sort of interpretation of this whole feedback story is that, you know, we might be on the wrong track, really. I mean, I think it might very well be just enhancing visual processing rather than being tied to conscious experience.
Okay. All right. Thank you.
Ellie, do you have an additional question?
Yeah, I more of a comment. These ideas about the separation of the hemifield or hemispheres, vision, etc., is sort of too strongly stated on both sides. And you know, I was just reading an old paper by Jampolsky in which he cited links, and he says it is inconceivable. This is about suppression in strabismus, in which there is a sharp line at the fovea between the two hemiretinas. It is inconceivable that such thing as sharp dividing vertical line should exist in the macula of the retina. Now, the funny thing about this is that the evidence for such line is overwhelming, and Lynch knows about it, and yet he says it's inconceivable that we could even that. It's just a guy in the zoo that looks at the giraffe and says there is no such an animal. So we have to be careful of that. Clearly, there is a lot of communication between these two hemiretinas, if you want. Evidence is overwhelming in my mind. There is starting with midline stereopsis. Midline stereopsis should not exist because it goes to two hemifield, but we have stereopsis in people that have split brains that had their chiasm split. So there is a whole lot of communication between the two hemispheres, and starting as early as a chiasm even, and certainly, you know, the question of feedback to the LGN, is it from the same hemisphere or coming also from the other hemisphere? I know it's tremendously large, but I don't know where it's coming from. So I think that the issue with many of these experiments is that no one should doubt the results if they're done reasonably well, and the carefulness should be in the interpretation of what does it mean, and we should be open to more than one interpretation, at least until it's get rejected. That many of these things. So what I'm saying is, a patient like GY says that he or shows that he can distinguish direction of motion. Well, direction of motion only takes low pass filter, and it could be many reasons to be able to do that with things that are far different than the idea that there is a vision-like sensation. So, you know, to distinguish left from right motion, all you need is low pass filter. For we showed it for almost 100 conditions. So it's not really.
Yeah, and he can't do the task if you do a stimulus where, you know, when you replace the dots on each frame, so
right,
but
yeah.
you can't track individual dots, so then he can't do the task. So in fact, you know, he can't do real motion perception.
And similarly with the completion thing, and I mean, okay, it's fascinating. But again, we got to be very careful. Even the very best experiments that have been done on that, and there's some really amazingly good experiments done with strings and and sticks, so to speak, remain problematic because it's the interpretation. To say that the person sees a full square you know, you need a really strong evidence to provide, to accept that interpretation rather than that he knows what a square looks like. Or, you know, there's many other things that they report seeing a face as complete. Well, you know, it's very difficult to think about that, and clearly they don't see the other half of the face. So how are they creating it? Is it really vision? It's got to be, what I'm saying is they report it, they see it, and then, as I said, some really great experiments have been done to show that there's no eye movement, no picking, no other things, but the interpretation goes a little too far. And so I'm just suggesting that we have to be cautious in the interpretation of these things, even though it's fascinating, and I like the interpretation too.
Thank you. Juha, there's in the chat, there's Gabriel Kehat is asking, we learned that people having blind sight can feel emotions and facial pictures. Can you explain this? Well, I assume the explanation is quite complicated, but do you have any insights about this? About understanding facial emotions in blind sight?
I mean, that's a complex. I mean, there's lots of work on this sort of other pathway associated with emotion processing, which I think has been research studies have shown that's not necessarily valid. But I think in lots of these things, when we talk about his awareness, it's a bit like I said before, I think you learn over time to kind of, you experience, I mean, when there's a visual stimulus and has a physiological response, you kind of somehow become aware of it. You know, it's a bit like those biofeedback stuff where somebody tells you, you know, can you modulate activity in MTP5 in fMRI scan, and you can do that. So you sort of have some sort of insight into your sort of physiological responses. But I think very often, you know, those are phenomena, those are not visual. And I think that's probably that sort of a phenomenon where you just, after so many trials and after so many experiments, something like GY can actually probably learn to develop insights into this sort of physiological responses. That's, I mean, that's one view of explaining these sort of things. But they're separate from qualia. You know, they don't have the same kind of experience. I'm gonna have to teach. I'm sorry, I'm gonna have to go because I'm teaching.
Yeah. Thank you very much again for joining us, and I'm sorry about all the technical issues.
Well, I'm sorry.
Really, I'm inviting
Thank
everybody to give a big applaud. It was a great and inspiring talk, and thanks for joining us.
you.
Thank you. Bye.